So this video will cover the Unit 2 third part of the Reproductive Biology section, which is on the biology of control and fertility. And this first video will cover the infertility treatments, and the second video after this will cover the contraceptive parts of it. So initially you need to know about fertility in general and what fertility means. So fertility is about the ability to conceive. So if you're fertile, you're able to conceive. Now conceive essentially means that you're able to have children. Now if you're infertile, then you're unable to have children, unable to become pregnant, or unable for fertilization to occur, depending on what the reasons are behind that. So in terms of male fertility, males are described as continuously fertile. Now what that means is that they are always they're always fertile, sperm are always produced. Now that will begin from puberty and then will last the entirety of their lifetime. Now obviously the quality of those sperm will decrease and the number might decrease, but generally the sperm are always produced. So continuously, just because they produce them all the time. Females are what's called cyclical. And cyclical essentially refers to the fact they're only fertile around maybe 3 days a month. Okay, so once a month, as only one egg is produced per month and one ovum is released per month in that process called ovulation. Now, in terms of this, you can describe the time that the woman is fertile as the fertile period. And like I said, that lasts for 3 days. Now those 3 days are the day of, the day before, and the day after that peak in LH. Now that peak in LH, remember, will be the day that ovulation occurs. So if you look at it on the cycle that we'd seen in the previous videos about hormonal control, this shows the female cycle. So very loosely, there's your follicular phase. First day being menstruation, and then the last day of the follicular phase being the day where ovulation occurs. So roughly day 14. Now the fertile period is that day of, which is the peak in LH, where ovulation occurs, the day before and the day after. So this here, 13, 14 and 15 on this female, is the fertile period. Remember that this can be different in different females. If they ovulate on day 17, for example, because their peak in LH is that day, then they would be the day of the 17th day of the cycle, and then the 16th and the 18th. In terms of pregnancy, what you would look to, if you were looking to become pregnant, for example, and they're talking about cyclical fertility for this female, you'd be able to detect that fertile period for 3 methods, or using 3 methods. So you can use this to monitor your body, and then if you were wanting to become pregnant, you'd aim for those particular signals and look for those signals for that. So for 3 methods, it's detected by a rise in body temperature. Now, although this isn't a massive rise, or it doesn't appear to be a massive rise, it is still quite large overall when it comes to body temperature. It's 0.5 of a degree. Okay, and that will remain that way for the remnants of the luteal phase. So the other half of the cycle. The cervical mucus will thin, and it will also become watery. And then the last one, which is sometimes can be detected through blood or urine, is a rise in LH levels. Okay, in either blood or urine. In terms of detection, you can also figure it out from the last time when there was a last menstrual period, but that's not exact in that sense because it could be different from month to month for a female. Okay, so these are all fertility parts, so these all describe male and female fertility, but the next parts will talk about what happens when they're infertile, or if they're there are maybe some certain issues within the males and the females. So what I'm going to do is I'm going to start with males. Now there are two different methods of male infertility treatments. Now the first one will be what's called artificial insemination. So the reason I've organised it this way is because the next one will be involved in one of the female processes, so I'll discuss that later. Artificial insemination is used when males have a low sperm count. Okay, and it basically means that they wouldn't be able to reach the egg, or very unlikely to reach the egg or the ovum themselves. So that low sperm count then, if the person has a sperm count is so low to the point where they're sterile, then a donor may be used. Now for this process, it's very key, it's very important to use very particular words. So they don't take several samples of sperm, specifically take several samples of semen. Now, to explain the difference between that, semen is both the sperm and the liquid in which they swim. Okay, so it's very, very difficult to actually just get single sperm. You would need to get a semen sample and then use that single sperm separately. So for this artificial insemination, you're using several samples of semen which are collected over a period of time, and then they're injected directly into the uterus. Now that doesn't mean that fertilisation will occur, there's no guarantee, but basically because it's going further into the reproductive tract of the female, it's much more likely they'll get to the oviduct and get to the egg that might be released. And this will be done around about when the time the female ovulates, just to kind of increase the chances of that. So in a similar vein, ICSI can be used. Now, this can be used if there is a low sperm count of the male, and also if the sperm are defective. That essentially just means that they don't work right. They might not be able to swim correctly, or their tails don't form correctly, or something along those lines. Now, with this, it might be useful to understand what ICSI stands for. Now, ICSI stands for intracytoplasmic sperm injection. Okay, so intra means within. Cytoplasmic refers to the cytoplasm, which would be here on an egg, and it'd be sperm injection. So if you break that word down, or that term down, you're involving sperm being injected into the cytoplasm. Intra means in, okay, or in towards. So in this process then, what will happen is the head of a single sperm, and it's very important to notice that it's a single one, it's not more than one, will be drawn into a needle, and then if you look at the diagram on the side, it's been injected directly into that ovum. So this here is an ovum. That here, that little guy there, is a sperm. Okay, so that's one single sperm being drawn up into a needle and then injected directly into the egg. Now again, specifying one ovum and egg, one singular one. You wouldn't do this in two sperm into one egg, or one sperm into one egg and then another. It would have to be one sperm into one ovum. Okay, and what that does then, that achieves fertilization, because then the sperm nucleus will fuse with the ovum's nucleus. Now this can then be done as a precursor, right, as the beginning of IVF, and that's why this is done in this order. So IVF is a female infertility treatment, but if males are also have a low sperm count, or if the sperm are defective, we can use this along with IVF. Okay, so I'll move on to the female one now. Now the two female infertility treatments are stimulating ovulation and then IVF, and both of these are linked. So ICSI can be used in IVF, and stimulating ovulation can also be used for IVF treatments. So initially, if you talk about stimulating ovulation, it can be used just in general to overcome infertility in females without IVF. So what they'll do is they can use drugs that prevent the negative feedback of one particular hormone. So negative feedback often is associated with estrogen. So drugs that prevent the negative feedback of estrogen on your pituitary hormones. And that pituitary hormone would be FSH. Okay, so if you then prevent that FSH secretion from being inhibited, that means the FSH will increase, and therefore an ovum will, a follicle around about an ovum will develop, and therefore it's much more likely to be able to ovulate later on. You can also get other drugs which will mimic the activity of FSH or even LH. So then that means that if you inject a person with these drugs, they'll have an artificial drug or a form of a drug that mimics the FSH and LH, therefore follicles will develop, and then ovulation can take place because LH levels are there to do that. Now, there's a problem with this in the fact that these cause what's known as superovulation. Now, superovulation is where there are a large number of ovum produced. Now, there's an issue with this in the fact that if you get many ovum, it's very painful and it can cause the ovaries to swell massively, and it can cause multiple births. So women that were trying to have one child end up having multiple potentially. But also those ovum that are produced could be used for IVF. So it can be useful. Okay. So this IVF one that we're going to lead on to will relate back to ICSI and stimulating ovulation at points. So IVF initially requires hormone stimulation to ready the female's body to produce ovum. Right, and that will normally involve injections of FSH. Now that then means that the follicles around an ovum will develop. Now once those ovum are developed but not quite ready to ovulate, or not about to ovulate, we will then harvest them. So they can be harvested, so that means surgically removed. Okay, so those ovum are surgically removed from the ovaries. I should probably state that it's the ovum that are surgically removed, or ova are surgically removed from the ovaries. Okay, be specific and tell them where they are removed from. People tend to forget this point and they lose the mark for this when it comes to an essay question. Now once those ovum are taken out, okay, of that ovary, which is this point here, they then have to be mixed with sperm to then achieve fertilization. Now the reason why this is called IVF is because it's called in vitro fertilization. Now in vitro means in glass. So essentially the really key point is here that it is outside of the female's body. So sperm are mixed with many ova, okay, that which are removed surgically, in the lab or in a dish or outside of the female's body. Okay, that's really important to be aware of. Right, and that then results in fertilization. Now at this point, if sperm aren't able to swim or they might be defective, then ICSI can be used in this place. Okay, so if sperm aren't able to swim to the egg, or they aren't able to fertilize themselves, then you can use ICSI at this point. Now when fertilization occurs, zygotes are formed. And these aren't then immediately injected into the female. Okay, they are then allowed to grow and develop. They are incubated. Okay, so kept warm and allowed to grow. And they grow to form embryos. Or at very least a ball of at least 8 cells. Okay, so that would be the minimum number that would be required for that to be implanted. Now sometimes in this process, the reason why families are using this is because they might have a genetic condition that runs in their family. So they can then use what's called PGD, which is pre-implantation genetic diagnosis. Now, they do this then to determine whether a child or a potential embryo, would then have a genetic condition. So, for example, a single gene condition, so a single gene mutation, or also it'd be used to detect chromosomal issues. So any abnormal chromosomes. Now those ones that would have any chromosome abnormalities or any single gene disorders would then be removed and they would not be implanted. And the viable embryos, the ones that are okay, without any of those issues, would then be transferred. Now again, we have to be specific about where they're transferred to. Now, if you think about this, normal process would occur down the oviduct, and then they would implant into the uterus lining, the endometrium. So therefore, these embryos here would then have to be transferred into the uterus to allow implantation to occur. Now this diagram I've got here shows this. So these are ovum, or ova, plural, right, and these are sperm. Now, in terms of numbering here, this point 1 would be where the eggs are removed from the ovaries, or the ovum are removed from the ovaries. So this would be point 1 here on the diagram. Point 2 is where they are then mixed together in a dish and then allowed fertilization to occur. Now ICSI again can be used at that point in time. The zygotes then form and they're allowed to grow and develop to allow embryos to form. So this is point 3. And then at this point in time, pre-implantation genetic diagnosis would occur. So these are embryos that would be formed. PGD would be carried out on this. Okay, so this would be number 4. And then the last one here is in the embryos, the ones that are selected and are fine with no issues, would then be implanted or allowed to be transferred to be implanted into the uterus. Right, so that's the run through of all the infertility treatments for males and females. Remember that IVF covers a lot of them. It covers the stimulating ovulation and it covers ICSI in males. Okay, the next video will cover contraception.